All right. Hello, everyone, and welcome to the OTS webinar series. Today, we're featuring two trainees, both of whom presented posters at the last OTS meeting. We have Daniel Knight and Bahareh, who will present on Aptamer research. And so we'll just get started right away. So Daniel, you want to take it away?
Can you guys hear me okay? Perfect. So hello, everyone. So thank you, Aaron and Ronald, for inviting us to talk today. So for those of you that don't know me, I'm Daniel Knight. I'm a second-year PhD student in the lab of Dr. Maria DeRosa at Carleton University in Ottawa, Canada. And today, I'm going to be talking about my PhD work where we're doing an aptamer selection for the low complexity domain of TDP43 to be used as a potential treatment in ALS. And there we go. So ALS, or amyotrophic lateral sclerosis, is a neurodegenerative disorder that is often characterized by a loss of motor neurons. And so just on the left here, we have what looks like a normal nerve cell. And so we have a cell body that connected through an axon is innervated into a healthy muscle. And this axon is insulated by this myelin sheath. And once sclerosis takes over, this myelin sheath begins to break down. And what ends up happening is the axon itself degrades, and you end up with this atrophied muscle, and eventually that muscle dies. And so, of course, this can start anywhere in the body, but you then run into problems when it hits your respiratory system and your circulatory system. So ALS is frequently broken up into two main types, sporadic and familial. And so sporadic, as the name implies, is really at random. They don't have a true cause for it quite yet, but there's a weak genetic link to it that they've begun to establish. On the other hand, familial is entirely genetic. It's all based on inheritance from previous family members. So sporadic does tend to be the most prevalent within the population by a long shot, up to 95% of cases, but familial is much more rare. Now, as I mentioned, because the pathology of sporadic isn't quite known yet, that does make current treatment options quite limited. There are a handful out there that do improve the lifespan by a marginal value, but nothing quite groundbreaking yet that I think the community is looking for. On the other hand, familial, because it's genetically linked, the causes are well established, and they're still establishing new genetic links to this day. There's still a lot of work to be done there. But because it's genetically linked, the current treatments are not really more abundant, but are definitely easier to develop. So this is just kind of a broad overview of some of the drugs that are out there. This is by no means a comprehensive look. This is just kind of a very bird's eye view of what's going on today. And so one thing I want to bring to everyone's attention is the heterogeneity of the targets that different people and organizations are looking at. Each gray box here is a different target. And so it really highlights that the disease is not uniform. It's very, there's a lot of heterogeneity in this illness. And so what's relevant for this organization, of course, are nucleic acid therapeutics. And so there's a few that are already well underway, not a few, actually multiple, that are well underway into clinical trials at this point. And so they do target different genes. So C9orf72 is a very common one. The FUS gene encodes the FUS protein, and SOD1. And so one drug that you may have heard about recently in the news is tofersen, and it's now actually referred to as Qalsody. And so this is an antisense oligonucleotide that is developed by Dr. Tim Miller, and the folks at Biogen and Ionis. So they published their phase 3 clinical trial results in the New England Journal of Medicine just a few years ago. And that led to an approval actually by the FDA in April of last year, which is obviously very exciting. And just a few weeks ago was approved by the European Union. So it just goes to show that nucleic acid therapeutics for ALS are really on their way. So as I said, about 10% of cases are familial, and within this familial umbrella, C9orf72 and SOD1 are the most common genes. About a quarter of them are still unknown and those relationships are still being established, but there are a number of other genes that have small implications as well. Also with sporadic ALS, they are establishing weak genetic links, but the main takeaway from this is that it is still very, very largely unknown, and there's still tons of work to do in establishing that pathology. And so one thing that they've begun to look at is the different, of course, different proteins, biomarkers implicated in pathology. And one of those is TDP43. So a study was done not too long ago that looked at postmortem tissue of both sporadic and familial ALS patients. And they found in up to 97% of cases that there were aggregates of TDP43. And so even though this relationship isn't entirely clear yet as to how it may cause pathology, the cause and effect relationship quite yet, there's definitely something happening with this protein. And it's actually not just unique to ALS, it's also in frontotemporal dementia as well. Nearly half of the cases have this related pathology as well. So TDP43 is a protein heavily involved in RNA metabolism and DNA transcription. So it has roles in splicing, transcription and translation, also in stress granule formation. But the part that I'd like to bring to your attention for our purposes today is this C-terminal domain here. And so this C-terminal domain, or the low complexity domain, houses the majority of associated mutations within TDP43 for ALS. So it's also an interesting familial target and not just a sporadic target. This region is meant to mediate protein-protein interactions, and because it's highly disordered, is highly susceptible to aggregation. And so there's strong theories that suggest that aggregation originating from this domain may be eliciting toxic effects, both cellular and animal models. And so in addition to that proven toxicity, one thing that we're looking to preserve using a nucleic acid-based strategy is to preserve the endogenous nucleic acid binding. So because this protein is made to bind endogenous RNA and DNA, we want to make sure that we're protecting those domains. So those bind at the RNA recognition motifs here. So we don't want any of our nucleic acid aptamers to interfere with the binding and the protein carrying out its role. Also, we could just as well look at the N-terminal domain, but studies have shown that its oligomerization are actually necessary for physiological function. So we don't really want to disrupt that oligomerization process, hence targeting this C-terminal domain. So there are a number of working theories for how TDP43 may contribute to the onset of ALS. And so this is just a broad overview of some of those theories. So TDP43 naturally resides in the nucleus. And at some point, there's an altered equilibrium that shuttles the TDP43 out to the cytoplasm. And once in the cytoplasm, there's a couple of different things that can happen. The first is it can phase separate. So it forms these phase separated droplets that can coalesce and concentrate the protein into these droplets. And from here, actually dependent or independent sometimes of phase separation, aggregation can occur. And so this relationship here is still being investigated. Most work shows that phase separation leads to aggregation, but there are some studies that show that the phase separation is not a necessary point for aggregation. One thing that is very well known is once we do have aggregation, then it can lead to the buildup of these intraneuronal fibrils, of course, which then lead to a whole host of issues within a cell, such as oxidative stress, and then eventually disrupted vesicle transport and axonal transport. So from a therapeutic standpoint, there's really two main areas that we can look here for intervention. The first being this relationship here. If we can block aggregation, then we can potentially block the downstream effects, the toxic effects of such aggregation. But even moving upstream just a little bit, we potentially looking at the mislocalization factor. If we can prevent this mislocalization in the first place, does that lead as a viable therapeutic interventional point? So the next question to answer is there a basis for this? Can DNA or RNA aptamers be used for inhibiting aggregation for neurodegenerative disease? And the answer is yes, absolutely. So even in our own group, Aaron, which I know led this study as well, was just published last week in the Molecular Therapy Nucleic Acids. We selected a DNA aptamer for alpha-synuclein, which is heavily implicated in Parkinson's disease. And we found great inhibition of aggregation, also colocalization within cellular models and animal models as well, and inhibition of aggregation in those models. So there's absolutely a basis for this type of technology. And so in terms of an aptamer selection for this protein, there's really three main phases that we want to go through here. The first is the monomer and aggregate preparation protocol. And this is probably the most important part. We want to make sure that because the protein obviously has this very strong propensity to aggregate, we need to make sure that there's sufficient monomer in solution for the DNA library to interact with. We're not interested in binding aggregates, we're interested in binding monomers to prevent that aggregation. So we need to make sure that there's ample monomer in solution for long enough to make that happen. We also need to make sure that the aggregation based on the conditions we choose is not inhibited altogether. Obviously, that would defeat the point of the selection itself. So once we establish those protocols, then we move into the selection itself, and then we'll characterize our sequences thoroughly in vitro and cellular models. So this is just a general overview of the selection process we plan to go through for anyone that may be unfamiliar. But we start with this initial DNA library of about 10 to the 12 or 10 to the 16 random sequences, and you incubate this library with your desired target. This incubation is typically meant to mimic the end application of the aptamer. So in our case, it would be under physiological conditions. Once we remove non-binding sequences, we're left with the target DNA complex. And then through a multitude of different techniques, you can isolate those bound sequences. So then you end up with only the DNA, which is then subject to PCR amplification. And then you get this enriched pool for your subsequent rounds of selection. And so this process can be repeated anywhere from 3 to 15 or 16 rounds I've seen of selection. It really just depends on the nature of your target, your application, the method you're using, all those different things. But in all cases, once the final round is complete, you'll send the final enriched pool for cloning and sequencing. So the first thing we wanted to do is establish the aggregation kinetics of the protein to see which set of buffer conditions as close to physiological conditions as possible would leave us with ample monomer for our library incubation. And so there's really two ways that we found in the literature this is done. The first is to dissolve the lyophilized protein in buffer and then treat it with a molecular weight cutoff filter just to remove any initial aggregates that may have formed, and then quantify that protein and then initiate the assay. The other way is just to dissolve it in water first and then filter this and then buffer that solution to physiological pH before beginning the assay. And so this is in phosphate buffer. We found this at pH 7.4 with 100 millimolar sodium chloride. And so we found this to give us the most ideal aggregation kinetics. You can see here that the aggregation doesn't start until at least 4 hours, which is fantastic. That is more than enough time for us to incubate our DNA library with. The endpoint of aggregation was obviously a little varied, but the main takeaway that we are happy with here was this 4-hour mark. Then we wanted to see if the filtration step was really necessary. We were struggling with recovery of the protein when we were doing this, so we lost upwards of 80 to 85% of the protein each time we filtered it. So we wanted to just evaluate the monomer distribution without the filtration step and see if we could still see it. And we could, which was fantastic. The only unfortunate part here is we obviously wanted to have an equal amount of protein, but because of the filtration loss, this was all that we had left. But it did tell us everything we needed to know. And so from there, that was just after 1 hour. So we wanted to see how long it would take for the monomer to completely disappear. And the destaining portion of these gels still needed a bit of work, but it did give us what we were looking for. And so this showed that we still had monomer up to 6 hours following dissolution without the filtration step. We figure it should be similar with the filtration step, but again, just because we lost the protein, it just didn't appear on the gel for us, which was unfortunate, but it did give us what we needed to know. And so the last thing I wanted to make sure was if the protein was phase separating, did it inhibit aggregation, since sometimes that has been shown to occur. And so we just did TEM at 0 and 6 hours, and that is definitely not the case. There's still ample aggregation happening after 6 hours. So we know that there's still aggregation to be inhibited. And so the strategy that we plan to use is gel recovery. So this has been a common technique used in selections for proteins that do tend to aggregate, where they can isolate the aptamer monomer bands. So this was done with tau protein, which is also implicated in neurodegenerative disease. And so here you can see it's very clear to see where the aptamer binds with the monomeric version of the protein. So they can physically excise from there and then proceed with the rest of the selection. So in conclusion, we found that the LCD lag time was about 4 hours, which is fantastic, ample time for incubation. And even though we did lose some to filtration, the filtration step did not seem entirely necessary, and we had monomer for up to 6 hours. So coming up, we have the actual selection itself. We just started a few weeks ago, so we're well underway with optimizing and troubleshooting that. And then we'll get into characterization, and eventually we'll evaluate the inhibition of aggregation in cellular models. And so there's lots of people to acknowledge for here, including our big DeRosa group. Dr. Maria DeRosa herself, of course, has been instrumental in this, and everyone in the DeRosa lab. We have a pretty good time in there, I think. And also Dr. Jim Shorter and Dr. Miriam Linsenmeyer are our collaborators at the University of Pennsylvania that have been incredibly helpful in getting this going for us, and their advice has been instrumental. And of course, my funding partners, NSERC Promote and Carleton University. And that's all. If anyone wants to reach out, this is how you can reach me, or reach out to Erin as well. She has my contact info. And thank you for your time.
Yes, thanks, Daniel, for this very clear presentation. So if you have questions for Daniel, please use the Q&A function. But of course, I can start with a question. So what always puzzles me, so I've been working on protein aggregation disorders for the last, no, I don't know, 15, 20 years. And what always puzzles me is the debate whether these inclusions or aggregates or inclusion bodies or whatever people call them, some people say, okay, these aggregation bodies are protective because, when these aggregates break up, it can be toxic and can have all kinds of consequences. But your argument is super clear because you kind of assume that these aggregates are toxic for the cell. So this is also the intervention strategy that you're trying to develop. So where is the preventive part in your story? Are we now very sure that these are only toxic?
Yeah, so you're definitely right. Within these diseases, it's not always black and white that the aggregates are entirely toxic, and it is the case for this disease as well. The main basis that I think people are looking to target is the presence of the inclusions and the aggregates in the post-mortem tissue. That relationship, the cause and effect relationship between the aggregation and the toxicity may not be entirely clear and not 100% understood quite yet. But what is abundantly clear is in the post-mortem analysis that there are these large inclusions. And so I think some organizations and researchers, and ourselves included, are definitely taking the approach of that even though it's not entirely understood how and maybe the necessary pathways to figure out that toxicity, that this is an overwhelming observation made in the disease, and so this is where our main target is.
Yeah, it's always. I also think that most of the time you also have there is a correlation between the number of aggregates and the disease states, for example, within post-mortem tissue, but also in the different kind of disease models. And this is where I wanted to go for the next question. So you kind of mentioned that you want to investigate this approach also in cellular models. So which cellular models are you thinking of? Because also when I think of cellular models, many of these models don't show aggregation, at least not when you have endogenous models. So are you thinking about overexpression models, or is it also a way to mimic this in models that express TDP endogenous?
Mm-hmm. Yes, the cellular model work is done with Dr. Jim Shorter. at the University of Pennsylvania. And so, of course, we haven't quite got there yet, but our plan is to use an overexpression model to evaluate this work. He's had phenomenal work over his 20+ years of experience in this field, evaluating the toxic effects of TDP-43 aggregates in cellular models. So he plans to use, I'm not 100% sure on the name. I don't want to misspeak, but it is an overexpression model that Dr. Shorter uses, and he plans to help us with as well.
Okay, that sounds the right way, right way to go. So the question in the chat from Lakshmi: since the aptamer is designed to bind monomers, will the aptamer work in patients that already have TDP-43 aggregation?
That's a good question, and we don't really know. There's definitely a field where you can, or a lane, I should say, that you can go where you can look at disaggregation, and that's definitely up and coming in this area of research. We personally aren't designing it that way. We do want to target the monomer first. I'm sure there is a way that we could alter the selection to do that, but for our purposes, it'll just be to bind monomer first, and then maybe that's an avenue we explore later on.
And to follow up, for most of these strategies, delivery is the most difficult thing, especially delivery in animal models or ultimately in human beings. So how to deliver this very precisely to the motor neurons?
Yeah, so we've had experience using a liposome-based delivery strategy. So in our Parkinson's work, we used a liposome that had a transferrin receptor aptamer that coated the outside of it. And from there, it delivered the aptamer very effectively across the blood-brain barrier in animal models. So we may look into using that strategy for this work. But in another breath, we're also looking at different ways to improve that delivery strategy in the Parkinson's work using different conjugates, chimera type related work. So depending on how that work goes, we may end up changing the strategy for this aptamer down the road. So it's something we've had success with the liposome work, but if we find promising results with other delivery strategies, we will adopt those for this for sure.
Yes, thanks. And so I think in one of your first slides, you showed all the therapeutic strategies that are currently into a preclinical or clinical trial. So it's a whole range from targeting specific processes to really targeting a specific gene. So what are the pros and the cons of your specific strategy, and how will it benefit all the patients? And also how it's related to all the other strategies.
Yeah, of course. So definitely the advantages with using aptamers, especially specifically DNA aptamers. DNA aptamers in-house, we can make them. So the synthetic process is obviously quite straightforward. We can modify them very easily, and because they're DNA, of course, they are inherently more stable in physiological environments. So that's one advantage for using this technology. Of course, because it is DNA, it's not inherently toxic to the body. So we do have that advantage as well, instead of like potentially a small molecule or something of the sort. Definitely some disadvantages for the aptamer technology is interference, because it's not a covalent interaction, it's all non-covalent interactions, then it is highly susceptible to interacting with the multitude of different proteins and other things within the cell and within the body. So finding an aptamer that is specific enough that can outcompete those types of interactions in a physiological system it's not easy. It can be done. It had been done before, but that's probably one of the main drawbacks to using the aptamer technology. It also if you look at it compared to something like an antisense oligo, the antisense is so specific because of the mRNA transcript it binds to. So that's definitely an area that the aptamers aren't as strong in compared to those types of technologies. But yeah.
How to design such an experiment that you can test the off-target effects?
Sorry, say that again?
How to design an experiment where you can test the off-target effects of the aptamer?
Oh yeah. So we use, at least in our lab, we can test using various concentrations of serum. So depending on the system we're going into, for mice will obviously be the first one. You can evaluate the binding in different levels of serum. You can include other relevant targets. So for this work, other amyloidogenic proteins would be a great starting place to evaluate its specificity. And because we're also targeting only one fragment of the protein, we can look at the other regions of the protein and evaluate the specificity to those. So there are a lot of options in evaluating the specificity of the aptamer. It just depends on the system you're looking at, but that's kind of the avenue we would go.
Yes, and then so I'm not sure the duration of a PhD differs in different countries. I'm not sure how many years this is in Canada, but I assume it's around the 4 years. So your second year? So what to do for the next 2 years? What would be your main goal to reach if you come back in 2 years and we have this conversation again?
Yeah, that's the magic question. Yeah, lots to do, of course. Once the aptamer selection is complete, then I think the immediate next step for us is to characterize in-house and then characterize in those cellular models with our collaborators. We're in discussions also with another collaborator about evaluating mislocalization. That's something we may end up exploring as well. So really the main priority is, it may seem obvious, but just getting the selection completed and getting our sequences. And then from there, it's binding affinity, it's inhibition effects, both in solution-based assays, but also in cells. And then all the other things, secondary structure, melting temperature, all those parameters that we want to characterize first. So I think it can be 2 years, but everything's a hypothetical, of course.
Cool. Well, thanks a lot, Daniel. Good luck with your PhD, and I give the word to Aaron.
Great. That was a great talk, Dan. Up next, we have Bahareh.
Can you see my slide?
Right. Yep, they look great. Go ahead.
Okay. So hi everyone. My name is Bahareh. I'm a PhD student in Dr. Drosa's lab, and today I'm going to present part of my results of my study, which is about characterization of acute myeloid leukemia, specific DNA aptamers. Leukemia is one of the most prevalent diagnosed cancer-related mortality in Canada, and acute myeloid leukemia is one of the main subtypes of leukemia with a poor prognosis. It's a type of leukemia that we can find in adults and children. Once we talk about cancer, we think about all the traditional methods that we have to fight with disease, like radiotherapy, chemotherapy, surgery, sometimes a combination of radiotherapy with surgery or chemotherapy with radiotherapy. The main question here is all these traditional methods, they have their own advantages and they have their own disadvantages. So, for example, in chemotherapy, we have to apply a drug, like sometimes almost double dose of the amount of the drug for the patient in order to make sure that the drug can reach to the target site. So this may cause organ failure in the future for the patient. And also, since we apply drug, these drugs can travel all over the body, and they can hurt cancer cells as well as healthy cells as well. They are technically the challenges here. It's a non-specific distribution of the drug. So we are looking for a way to make this more specific and to address this challenge. I'm going to introduce Aptamer as the powerful ligand. Aptamers were first described in 1990, and the word aptamers comes from two words, aptus and meros, so fit into the part. And they are short single-strand DNA. They can be RNA nucleotides that binds to the target with high affinity and selectivity with a unique three-dimensional interaction. So the whole idea of my project was to develop the aptamer for acute myeloid leukemia disease. And in order to reach this aim, we had technically 4 goals. We prepared and characterized the aptamer sequences against leukemia cells. Then we tried to identify and validate the potential biomarkers for the acute myeloid leukemia cells. We try to make 3-dimensional spheroids of the leukemia cells to assess the localization of the aptamer and to see how our aptamer is specific towards the leukemia cells, not the other cells. And we will do the biodistribution and accumulation of the aptamers in NOD mice with in vivo imaging system, which is called IVIS. So the SELEX method that our lab did actually in order to select the sequences of the aptamer, we will go through SELEX method. Our lab, this method, this SELEX method has been performed already in our lab. And I think they did it in 2018. I wasn't part of this, but the way that they select the sequences was we had our target, which was the AML cells. We incubate the target with the random library that we had. And then for the counter selection, they chose fibroblast cells, WI38, which is the human lung fibroblasts. Once they washed all the non-selected binders, they ended up with the selective binders, and then through the PCR method, they ended up with 3 different sequences that they named them aptamer 1, 2, and 3. Because of the double hairpin structure of the aptamer 2, at that time, they decided to choose this aptamer as a lead aptamer. But when I started my project, I decided to work with aptamer 2 and aptamer 3 because of the low KD of aptamer 3. And then once our lab chose aptamer 2 as the lead aptamer, they did flow cytometry against the leukemia cells and also against the fibroblast to see the binding affinity and specificity of our aptamer. So here you can see the pink sheet, which is happening once we treat the cells, the leukemia cells, with the aptamer number 2. And once they treat it with the fibroblast cells, you can see the blue sheet for this peak is happening, but it's almost negligible. And it's happening because of some type of non-specific binding, but in compared to the like actual AML cells, it's almost negligible. So once I started my project, we decided to see actually where the aptamer binds to the cells. Are they going around the cells? Are they gonna internalize through the cells, through endocytosis, or are they gonna be like some type of surface biomarkers that the aptamer binds usually? Because our lab did SELEX against the whole cells. So we start to discover this, and to answer this question, once we culture the cells, we treat the cells with the aptamer, both aptamers, and their scramble. And also we had another control because we also use the DNAs. After the treatment is done, we added DNAs to the treatment, since the DNAs can show up all the aptamer around the cells, and my also aptamer is a DNA strand, single strand DNA. So this could be, without this could be a good control as well. And then we visualize them under the fluorescence and microscope and confocal microscopy. And I have to mention that my aptamer has fluorescence tag. So here is the results that we got. After we visualize them under the fluorescence microscopy, here we stain for nuclei or with Hosh, and also the green color shows the aptamer. The left image is aptamer 2, the right image is aptamer 3. As you can see, some punctate localization of the aptamer is happening. So the binding is kind of interesting for us. And also we got similar results for aptamer 3. And I just wanted to share the happy face here that I got for aptamer 3 as well. So we also visualized this with confocal microscopy, and we got similar results, like some punctate localization for aptamer 2 happening at the specific site of the cells. And also we saw some internalization of the aptamer into the AML cells. So we start thinking of, would it be like some type of organelles that might taken up the aptamers or not? So to answer this question, we start to stain for organelles, and we start to stain for endoplasmic reticulum. So once we did the treatment with the aptamer, we start treating them with the endoplasmic reticulum tracker, which has the blue color. And then we fix the cells with paraformaldehyde and visualize them under the confocal microscopy. And also we had our control as well. So here is the results that we got. Here, blue stands for the endoplasmic reticulum, blue color, and green is our aptamer. In this specific experiment, we decided not to stain for DAPI to avoid overlapping of the color, blue color. So we conclude these images and we start to think about that. So we got nice images, nice microscopy images of fluorescence and confocal, even for internalization. But what about the punctate localization that we saw during the microscopy? In order to answer this question, we did pull down assay in collaboration with NRC in Ottawa. This time we used our aptamer. We tagged them with biotin, and then I did this experiment in vivo and in vitro. What I mean by in vivo means I used it against the whole cell, and in vitro means I lyse the cell, and then I treat them with the biotin labeled aptamers. And I used streptavidin magnetic beads, and because of the high affinity of the streptavidin towards the biotin, we were able to catch the potential biomarkers, and then we released the target proteins. We ran gel, and then we identified them by the mass spec. Since I'm still working on this part, and we got our potential biomarkers, I can't share it in this presentation, but we got some potential biomarkers that we are hoping that this might be our biomarkers. And then the next chapter of my experiment is starting with one question: that do AML cells form tumors outside of the bloodstream? We know that in the patient body, AML is in the form of mostly cluster that can travel all over the bloodstream. But Can they make the tumors outside of the bloodstream? And the answer of this question is yes. Myeloid sarcoma is a tumor of myoblast or immature myeloid cells, which is occurring in 0.7 per million children and 2 per million adults. This tumor presenting mostly in bone, soft tissues, and lymph nodes. So therefore, we start to thinking about that if we want to make our like a model of the 3D model of our AML cells, how we can do that first of all. So in order to explain better and have a better idea of how we can make this, let's look at the 2-dimensional cell culture and 3-dimensional cell culture. In 2-dimensional cell culture, like the normal cell culture that we do in the lab, that we use normal flask, we have a monolayer cell that we usually culture them in the media. But once we move forward to the 3-dimension, we usually use low attachment round plate that we can provide the culture media, and also we should provide extracellular matrix for the cells in order to make a nice spheroid for the 3-dimension model. So technically in 2D cell culture, it's we can say it's a very easy technique. It's very convenient that you can just use it very easily. But in 3D cell culture, which is representing the tumor microenvironment better than 2D, and also we can increase the cell-cell interaction to the extracellular matrix, and also it's more applicable drug resistance model. Therefore, we decided to make this 3D model of our AML cells with the help of fibroblasts. This part of experiment has been done in collaboration with Terasaki Institute in Los Angeles, and so we used ultra low attachment spheroid microplate, and we used co-culture system. Technically, we co-cultured AML cells with HEL cells, HEL299 cells. And the reason that we use fibroblasts is 3D fibroblast co-culture have been used to mimic the 3-dimension tissue orientation and architecture of extracellular matrix environment, microenvironment in vivo more closely. So the way that usually co-culture system works is, so we have our AML cells separately and we have our HEL cells separately. Since we had to determine the amount of the cells, it depends on the amount of the cells, the ratio, like which ratio we want to go for each cells. So we had to go through different steps. We had to go through different number of the cells at various ratio. And then, so we had our control, AML, and then we had our HEL as another control. And we have our scramble as well for the 2 aptamers. And also, for example, once we wanted to culture aptamer versus like AML versus HEL for 2.5K, the total number of the cells, we had to go through various ratios like 8:1, 16:1. That was the best ratio that we got after we did our first experiment. And also, we tried different number of the cells for each ratio. So here is the images that we got after we co-cultured them. As you can see for the AML cells, which are the non-adherent cells, it's not possible to make the spheroids even in the low attachment plate. So as you can see at day 3, even at the low number of the cells or a higher number of the cells, we can see some clusters. And here, in order to know and have a better understanding of which cells is AML and which cells is HEL, we decided to use a deep red cell tracker for AML. So here the pink color shows AML cells. And if you look at the HEL cells itself, so we didn't stain here the HEL cells, but you can see the spheroid formation happening for the HEL cells itself. But once we go through the co-culture system, it's kind of interesting because so you can see that once the majority of the cells is AML versus fibroblasts, we can see some clusters of the AMLs, even though if we go to the low number of the cells. But once the majority of the cells is fibroblasts versus AML, we could get the spheroid out of the at the bottom of the flask, at the bottom of the plate. So, and also we played with the various number of the cells, and also various ratios like 8:1, 16:1, 4:1, 2:1. We tried different ratios, and here, like at the ratio of 16:1 for this number of the cells, we got a nice spheroid for HEL versus AML, and also for 8:1 for this number of the cells, for HEL versus AML itself. And then once we got these images, we decided to treat them with the same aptamer that we treat in the two-dimensional format, just to see is it specific to our aptamer or in our actual 3D spheroids, which is sometimes happening in the patient body. It's going to be more specific, or it might be some non-specific binding. We wanted to answer this question. So once we treat them with the aptamer, this is aptamer number 2 that has been treated with, this is AML, has been treated with aptamer 2, and this is aptamer number 3. This is our control, AML itself. And as you can see, this scramble for aptamer 2 and aptamer 3. The green shows my aptamer. The blue, it's just the staining that we did for nuclei, it's Hosh. And the pink is just my AML cell. And once we move to the HEL cells, we didn't see any binding. So keep that in mind once I move to the flow cytometry data. And then once we did co-culturing, like once we had the majority of the cells was AML, we can see some sort of binding is happening, like green, which is our aptamer. And we can see also some similarity towards the two-dimension confocal microscopy that we did, like some sort of punctate localization that's happening next to the pink cells, which is our AML. And we repeat the experiment. So technically, we took the image for the spheroid once we had HEL as the majority. And here we can see that the intensity of the fluorescence, it's more intense, the green fluorescence here. So we were start thinking that, is that possible that some HEL cells also taken up the aptamers as the non-specific binding, similar to the way that happening once our lab did flow cytometry versus the WI38, as I showed previously, like some slight changes in the pink, not in the pink, in the blue shift for the WI38. So we started thinking about that. And then we decided to do flow cytometry for a spheroid formation that we did. This is the image. This is the results of the flow cytometry for two-dimension. Like once we had two-dimension studies, once we treat the cells with the aptamer 2 and aptamer 3, as you can see, it's very significant rather than HEL cells for both aptamers. And also we did that for a three-dimension study, like for three-dimension spheroids co-culturing system that we did. And here you can see that through the co-culturing system, once the majority of the cells was HEL, we were able to see some intensity, as I show in the images also. So this is telling us like maybe also some HEL cells is sharing some sort of common biomarkers with our aptamers. We will do cool down against the HEL cells and against the WI38 cells with the same aptamer that we have. and probably we are hoping for like coming up with the same or maybe common type of biomarkers and then specific biomarkers towards the AML sites. So that's the plan. After we got the flow cytometry and confocal microscopy data. So the next step of my experiment will be technically validating the proteins that we found so far. And we are guessing it might be the surface protein that we are looking for with Western blot. And also in collaboration with Terasaki Institute, we will do IVIS in order to see the aptamer biodistribution in nut mice to reveal the accumulation of the aptamers at the tumor sites and also in other tissues. So my big acknowledgement goes to Dr. Drosa and Dr. Wilmore's lab and all my friends at those labs, Dr. Twine and Dr. Chen at NRC, Dr. Ernest, Dr. Mulai, and Dr. Marino at Cincinnati University, Dr. Khadem Hosseini and Dr. Hassani at Terasaki Institute. And of course, Carleton University promotes for funding the project. And I appreciate the time that OT is giving me to present my data. So I'm also available. Feel free to ask me any question you have. Here is my email address and my LinkedIn page. Since I'm finishing my PhD soon, I'm looking for the postdoc, like industry job. And I would be glad if you can send me a message if you have any questions. Thank you so much.
Yes, thank you very much for this presentation. So if you're looking for someone, please contact Bahareh. But of course, let's start with some questions. If you have a question, please use the Q&A function again. So when I was looking at your data and you showed some images of your 2D culture system, and you showed the localization of Aptamer 2 and Aptamer 3. And when I looked at the images, for me, I thought there were already some differences just based on the localization and the presence of the green signal. So did you ever quantify this just by some easy ImageJ analysis where you look at co-localization between the nucleus and the green signal of the aptamers?
Not yet. We know that the two aptamers are different. We know that we're going to end up with two different biomarkers for two aptamers. But for both aptamers, if you look closely, for both aptamers, they're going to bind to the one specific site for aptamer 2. Aptamer 3, we saw some internalization. That's why we thought it might be some organelles that binds through the aptamer. But through the pull-down assay, since just the pull-down can answer us this question, like macroscopy is always good, but it's better to go through flow cytometry and pull-down. Through the results of the pull-down, which I couldn't share, we can say that aptamer 2, it's a surface biomarker, and aptamer 3, it might be some organelle. The pull-down assay kind of confirmed this for us, but I'm still repeating pull-down and I'm still getting data. So that's why I couldn't share that. But yeah, there is a change. There is a change between two aptamers because they are two different aptamers, two different structure, two different KD. It should be a different behavior of the two aptamers.
Okay, so well spotted. So are you planning to so I think you did some co-localization experiments with the ER. So are you planning to do some co-localization experiments with other organelle markers then? Or with some surface markers?
We did with Golgi, but we couldn't get a nice image once we tried Golgi organelle. But that's all we could think of, because it's like, if it's secretory protein, it must be either in the plasmic reticulum or Golgi. And we are thinking about secretory protein is coming from ER and Golgi. But we even optimized our Golgi staining protocol for a couple of times, but we couldn't get a nice image that I could put in my presentation.
Yeah, and I also noticed that you were not taking along any nuclear marker. So has that a specific reason? Because it might be nice to also take along so that you have a bit.
No,
at least
no, the reason is once they did SELEX, they did it against the whole cells.
Ah, okay.
It should be surface biomarker.
The other thing is, well, you switched from your 2D model, you switched to the 3D model because that has all kind of other advantages. Are there any? So maybe both for 2D and 3D. Are there next to the co-localization experiment or the imaging experiments, are there any functional outcomes that you can test? So is there like an assay that you can do and you can measure something in the cell apart from fluorescence?
Not for 2D and 3D, but for animal study, we're going to collect the blood of the animal, of the mice, and we will do flow cytometry for the animal study. And then we will do histology also for the animal study, just to see the co-localization of the aptamer, where it's going to be accumulate. Like we're going to do some histology for like different organs, like brain, lung, kidney, liver, and we just want to see where the aptamer accumulate once we test it against the animal.
Yeah, that sounds like it. Yeah. But not in the 3D. Yeah, the 3D is also because the 3D is not. The 3D is a clump of cells, right? It does not really have distinct structures within the spheroids. So, or does it have? I'm not too familiar with it.
No, the 3D model, it's like a exciting area. Like it's coming from the tissue engineering people, because once you have a 3D model of your tumor, you can say that it's mimic my actual tumor. That's what is happening in the patient. Like all the tumors in the patient body, it's like 3 dimension. They are not 2 dimension, and that's the reason that most of them, once you apply drug, the drug can't reach out to the actual cancer, or you fail. Like pharmaceutical company, usually they fail once they apply the drug, because it's 3-dimension format of the solid tumor, and usually it's chemo resistance once you move forward to the 3D. And that's the way that usually people make this 3D in the lab in a small case and test the drug on that, just to make sure that how it really responds if they apply it like to the human later, like the drug. That's why we thought, so maybe if we can make the 3 dimension of AML, first of all, it's very interesting. So I couldn't find any paper through the co-culture system of AML with fibroblast that they could make the spheroid of the AML cells. So this is new. And also the way that actually worked through the microscopy images. And so the way that we chose fibroblast that was the reason behind that. So we just wanted to know how specific our aptamer is, because our aptamer has been developed against AML cells. So technically it shouldn't bind to the other cells, but always there is like some non-specific binding that we can't avoid that.
Yeah, and maybe to add to this, I'm not familiar with this tumor model, but I'm familiar, for example, with the brain organoids, and one of the avenues that they wanted to go is to also include the vasculature in these. I think these are slightly bigger organoids. So is it also an approach that people are going for the tumor models, that they want to add vasculature so they can study that as well?
I believe so. Yeah.
Because that would be pretty relevant for these kind of models.
Yes, but for my project, like if you look at like my data, like my AML cells and everything, people usually work in 2D for AML cells, because in patient it's like a cluster. Like they never think about like 3D model. So more 3D model for blood is kind of it's a big question. Like blood, it's like 2D. There is no 3D for this. But since there is a disease that exists, so it's better to always look at the 2D and 3D for the cancer study.
No. Cool. Before I finish, do you want to pitch yourself for half a minute? That you say, what is it what you are looking for the for next job?
I'm looking for like postdoc right now. I'm looking everywhere, like industry job or even university. But my ideal job would be a postdoc for now.
Okay, and any specific areas or specific that you wanted to go for?
Cancer drug delivery.
Okay, so if you are looking for a postdoc on cancer drug delivery, then please go to Bahareh. So, Erin, I give the word to you.
Great. Well, those were excellent talks. If you're interested in Aptamer research in general, or you're interested specifically in the topics of Dan and Bahareh's talks, please do reach out. You can do that through the info@oligotherapeutics.org email address, or you can contact them directly. We do have some webinars that are coming up. So July is pretty full. We have one July 11th, 18th, 25th, and then at the end of August. So make sure you do check out the webinars website, off of the OTS main website. You can find out all the information about the speakers there.